This talk is about technology and data analysis. I intended to keep my talk a little bit different distance from science and biology for two reasons. First, I'm not a good scientist, especially not an up-to-date biologist. Second, lots of scientific data I referred in my talk were generated from my collaborators and the students who worked with me. Those data haven't been published yet. That's why I hesitated to touch too much with those data. I know you may have questions about those data and the result, but you may not be able to get a good answer from me because of this hasn't been published. So talk today is about data downstream analysis, next generation data downstream analysis. Can you hear me? Okay. So first question you may ask, what defines downstream and upstream? I would say everything starts from FastQ file will be downstream, because above that, it's really an automated machine process. You don't even know what is that. Why do you care? So then question is, what is FastQ file? The FastQ file is output from sequencer, which consists of millions and millions blocks. But good thing is, this file is human readable. It is a friendly text file. Each block has 4 lines with header, the header line, the first line, the second line followed by sequence read. That's the DNA sequence. Then third line is the repeat of the header line. In the new format, this line has been changed a little bit. The fourth line is the quality assessment score. I will have more to say about this line later. So the first line, header line, begins with a sequencer identifier. The sequencer identifier, which was hard-coded in the Illumina machine. In the old time, Illumina produced the sequencer distributed to all those genome centers. In order for them to do sort of surveys, they have this unique identifier for each sequencer, just like a VIN number for your car. So follow this identifier, the number is the lane number. When you do your experiment, you put a sample onto that flow cell. Each flow cell has 8 lanes. This number tells you which lane is that. Then followed that is tile. What is tile? Tile is a bunch of pictures created by fluorescence camera within the sequencer. So on these tiles, you can see lots of spots. Those are sequence read. Each time, the sequencer camera moves to a certain area to create a picture, which is called a tile. Followed tile is the coordinates, which identifies the sequence read. So after that, you will see the number sign and the index of the multiplexing, and then the paired end information. In the new format, you will see a slight change. The header line, you don't see those vehicle identification number anymore, but instead you have the HiSeq or GA, some type of identifier. Follow that header, the first part of the headline, it's a round number. What does round number tells you is so important to me at least. I believe this number tells how many times the sequencer has been used for sequencing. This will help you to do sort of estimate if the sequencer machine needs to be maintained. For example, lots of time, the optics camera in the sequencer need to be recalibrated periodically. This number will definitely help you to identify that if your machine needs to be recalibrated. Then, followed that is flow cell number. This is a log number for flow cell, just in case some type of technical issues come up with your result. So they can trace back what log of the flow cell did you use for your experiment, if this is your problem or manufacturer's problem. So after that is same lane number, tile number, and the coordinates for each spot. And then here, there is slight change for paired end information. And then the N means this read passed filter. If it is Y, that means that read failed. Then control bits, I don't know what's that for, but the last line is pretty much important. That's barcode. So in the fourth line I mentioned earlier is the sequencing, the quality assessment score. So here you don't really see a score, but you can see lots of numbers and sign of keyboard. This is actually ASCII code. What you need to do, you need to translate this code back to the decimal number. For example, here letter H means if you translate the letter H back to the decimal, that's 104. This 104 equals to Phred score plus 64, which means if you minus 64, you will get 40. That 40 is Phred score. So in the old time, what we need to do is code yourself, calculate all those Phred score for each base, and assess all those reads. Now, you can download nice Java-based software to do this work. It's very convenient. However, all these are based on the Phred score. What is the Phred score? Phred score is a quality assessment score for each base. That's easy to say. But it actually is the probability of the incorrect base calling. What does that mean? For example, here you can see A. Phred score of this A is 10. What does 10 mean here? Which means probability for this base as an incorrect base is 10 in 100, or 1 in 10. In other words, the accuracy for this base is 90%. If a base you have with Phred score 20, which means the probability for this base to be incorrect is 1 in 100. In other words, the accuracy for this base is 99%. You might think 99% accuracy is pretty good, but in practice, you will find this way below the average bar. Why I'm saying that? In high-seq platform, usually you will get about 200 million reads per lane. If each read has 50 base pairs, you will end up with about 10 billion base pairs. If average Phred score is 20, which means you will have about 100 million incorrect bases in your file. That's absolutely not acceptable. Then you may ask, what Phred number will be acceptable? I would say it all depends what type of work you are doing. In my data analysis, if I'm doing SNP discovery, I would throw everything away which Phred score is below 30. What does that mean? That means the incorrect base will be 1 in 1,000. You got 1,000 bases, you have only one chance to be an incorrect base. Okay? If you are doing CHIBI, you can lower that bar to Phred score around 25. That's usually I'm doing. You can disagree with me, but that's really arguable, okay? So with all these knowledges, and then you can move on to do some type of work. With this FastQC file, next step, you will do mapping. What does mapping do? Mapping is trying to assign each read with the genomic coordinates. Okay? So you put this FastQC file with the proper genome you are studying, and with alignment tools. All the time we just use BLAST. Now you can use Bowtie, B1, that's much faster alignment tools. And then turn it around. You will get lots of output files, such as sum, bump, or bed files. What are these files? These files has no difference with previous one, but all those reads has been assigned with genomic position, okay, coordinates. So user definitely can take this file away to do some trick they want to do. For example, they can display these files in the UCSC genome browser or any type of genome browser, like G browser, IGV browser, and so on and so forth. Louisa take this file away to check if her knockout experiment is efficient or not. But usually a biologist has much bigger ambition. Okay? They want to know everything in the genome. They don't just stop their work here. And these are basically all those tasks biologists would like to do. Okay? They want to do some type of assembly for novel genome or novel transcriptome. They will do some type of study for DNA replication or damage repairing type of study. All these works we have done. But because of time, I do not have anything cover these two parts. And majority of work come to me are ChIP-seq or RNA-seq. What is ChIP-seq? Basically, it's positioning genome elements and define some functional relationships. And RNA-seq basically is to detecting the productivity and the frequency of the genome product. So in order to accomplish those tasks, you need to have a little bit knowledge about the genome. What is genome structure, and how does genome process the information, and so on and so forth. Here is usually a genome look like. They have a DNA sequence wrapped with histones form a nucleosome. A nucleosome then forms the chromatin. The chromatin finally becomes chromosome. The information process is backward. When a transcription factor comes to the nucleus, it will bind to a certain area of the genome, trigger some type of function, like DNA replication or transcription. These are basically basic genome functions. Okay. DNA replication, DNA repairing, and transcription. The transcription event is a centerpiece of the genome function. So to review this event, I have a little movie here to help us to get more insight knowledge about the transcription. So I hope you guys can enjoy this, and I can take a 2 minutes break.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Okay. From this movie, we have learned something about the genome. In the genome, there are lots of genome elements, such as promoter, enhancer. I would say most important genome elements is transcription start site. Almost 99% of studies come to me is studying a protein which binds to a certain area of the genome. People want to see how far or how near this binding site to the gene transcription factor, gene transcription start site. So besides the genome elements, there are also lots of landmarkers within the genomes. For example, the histone markers, and also the DNA methylation site. All these genome elements and the landmarkers are so important for your research project. Okay. So most of the time, when people are studying their genes or their proteins, they would like to know how really coordinate between the protein they are studying and these landmarkers or genome elements. Here, what we did is trying to find out what type of histone modifications are really on this transcription start site nearby. Okay, so we can see the histone marker K4ME1, K4ME3 almost stick with the histone, the transcription start site TSS. Okay, others is a little bit fuzzy, doesn't form a very clear line. So some other time, people would like to know some average distribution of the read. So to do that, this type of work is really help for people to understand broadly what is their protein doing. So how to do that? Okay. First, we need to calculate the read density base by base and normalize that against the library size. Then we define the region of interest and plot that normalized rates distribution in that region. Okay. But I need to point out is that this type of plotting, so raw rates plotting, is semi-quantitative, okay, which means you can look at this data, but if you want to really believe the quantity difference between different samples, you need to do more, because variation, I believe, which inherit from your sample preparation. Let's say you are preparing your chip samples with sharing method. It will end up between 200 to 2,000 base pairs. But in different sample, for same region, it can be longer or shorter or longer DNA pieces. When you do next PCR cycles, it will increase this difference. So that will probably hurt you make a judgment if this quantitatively higher or lower. Okay. So when you did these rates, distribution plot and so on and so forth, next step you would like to call rates enrichment area, which means everybody knows it's called peak calling. So peak calling can be done with lots of different softwares. I do not introduce this type of work here because we didn't really focus our work in this area. You can find tons of software to do this work, but keep in mind, none of those is perfect. After peak calling, you can do sort of work to correlate different peaks to see how far or how near they are. In this study, we would like to know the DNA replication forks. If this protein has something to do with the DNA replication forks, if it is near overlapped, we will probably have sort of idea this protein has something to do with DNA replication. But functionally, we don't know. This gives you sort of idea physically, how do they interact. And also, you can specify the distance, a fixed distance between the proteins you are studying and also landmarkers or genome elements. In this case, we would like to know if this protein has physically located in the same area as other histomarkers. We find out this protein has fairly well overlap with the histomarker K27 trimethylation. Okay. So this is basically what we are doing for CHIBI. Here is the summary of the CHIBI data analysis procedure. We start from, of course, FastQ file and do quality control. This quality control is sequence level quality control. And they map that to proper genome. And we do reads, distribution calculation, normalization, and so on and so forth. And then we do the CHIBI calling. So here are the results. If you work with me, you should expect to get this output. You will get FastQ file, of course, that from you, and I will send this back to you. There will be quality control output. Okay, how good is your experiment in the sequence level? And you will get these processed data files, some files, BOM files. You take that for visualization if you want. And you will get peak calling and annotation result. And for these nice pictures you will have, you can use that for your publication, grant application, and so on, for your thesis if you want. But one thing you don't really expect to get from me, that's differential peak calling, because I believe there are huge variations. You can't really trust that result, okay, unless you do further verification if you want. So now we are moving on to another big topic, RNA-seq. RNA-seq, I believe, is much more quantitative measurement for your genome-related study. Because it is quantitative, there are lots of issues as well. The first issue I would like to discuss is duplicate reads. The duplicate reads are artifacts created by PCR. Okay, if you have more PCR amplification, you tend to have much more duplicate reads. Here, what I showed here, this is the same sample with or without duplicate reads removal. You can see without duplicates removal, everybody lifted. If this lifting is proportional and linear, it won't be a huge problem. However, if you do the correlation plot, duplication removal, duplication kept, and the duplication removal, you do this correlation plot, you always see a group of reads which off transline. That's why when I do the RNA-seq analysis, first thing is to remove all these duplicates. Second issue is gene length adjustment. I know lots of people are doing gene length adjustment. My conclusion is gene length adjustment is totally unnecessary. Let me tell you why. And here is a table which show you the genes and the length and the raw counts and after length correction. You see, for the short length of genes, to get a reasonable FPKM, that's adjusted number, you only need 5 reads. For a gene, around 1,000 base pairs, to get a similar RPKM, you will need much more reads. What does 5 hands mean? If your library size is 5 million, which means you have 1 read per million. In current Illumina platform, it doesn't have such a resolution to find out a transcript within 1 read per million, 1 transcript per million. So that's very possibly a false positive reads. Okay? This, I know, this can't really convince you. However, if you do the gene length conversion, here are 4 different samples. Let's take a look. Let's focus on this picture. On the right side, these are plotting with the raw reads against the gene length. You can see lots of reads are piled up in this area. It's reasonable. Some of reads are really like gene length related. But if you do the gene length adjustment, you see all those numbers are associated to the short length of genes. Okay. I believe this correction is biased to the short length of genes, but I know this can't really convince you. The gene length adjustment is unnecessary. I will have one more slide later. I believe after that you will agree with me. Gene length correction is unnecessary. So, third issue is reads assignment. This is a very, I would say, not wide known issue, but very important issue, because in this room or somewhere else in the academia, lots of people are using top hat protocol. Okay. Here, what I did, after reads mapping, you need to assign those reads back to the gene model. Which software can give you better result for assignment? What I did, I compared 4 different assignment methods with simulated reads. What does simulated reads mean? I randomly picked 1,000 genes. Okay? Within these 1,000 genes, we have 20% isoforms. And we simulated those reads, so we know how many reads from one gene. That's we called true reads. Then I use different software to map these reads, assign these reads back to the gene model to see which software can give me best result. Here, you can see that the BIWA and the Expresso, this pipeline gives us the best result, which means the reads has been signed to the genes are really correlate to the true reads. The bow tie has a little bit of difference to the BIWA, which is understandable because bow tie doesn't really deal with insertion and deletion. Okay. Here is my method. I use this method plus the bow tie. These give us a similar result to the B one. The top hat cuff link protocol is worst one, okay? It's almost really didn't find who is who. That's interesting. You may want to know why top hat cuff link doesn't give you true reads. This is a reads assignment scenario, okay? So most of case, genes are really along with this type of gene model. One read per gene. But there are about up to 35% of reads fall into this type of scenario, which a read can be shared by different genes or gene isoforms. The way I did is based on the previous information we get. For example, in this case, we have 10 reads total. 9 reads can be assigned to the gene A without any question. 1 read will be assigned to gene B without any question. That ambiguous read will be signed based on this 9:1 ratio. What cuff link does, or top hat does, they don't do this probability calculation. They just split those reads, whatever left, throw that into your gene model. That's why you can see the results over there. So by this point, you may say, oh, I still get some good interesting result from top hat or cuff link. What can I say? That's really good for you. But there are basically lots of false positives with the top hat cuff link. Okay, just keep in mind. Here is another issue we have discovered recently with the barcoding. Okay. What do we have done here? We have barcoded samples and control samples. The blue are biological replicates. The red here and this one are exactly technical replicates, which means this sample has been split into 2 halves. 1 half with a barcode, another half without a barcode. Theoretically, if barcode doesn't have any problem, this sample will stick with these groups because these groups are same tissue. This group separate because they are different tissue. Okay. The variation here you can see basically generated by barcode. Then I had an argument with someone who says, okay, this is ridiculous. It can't be this way. I said, well, from the data, we can see this. And I want to prove what's going on with the barcode. So I took further look with the data. What I did, I correlate the barcode samples based on the genes, based on the fold change of those genes. I see all those barcoded samples has a very nice correlation, which means no matter what type of sample or what type of barcode, they always give you semi-exactly amplified genes. In this case, samples with or without barcode, they don't have this type of correlation. This is absolutely a hint that barcode in this experiment, maybe not yours, but in this experiment, gives you huge variation. Okay. So far, we are still doing some type of testing. I can't tell if this barcode problem is widely seen by other people. But I did have a communication with Illumina. They said, if you barcode samples with 5 or less, you tend to have this type of problem. Which means if you have much more samples to be multiplexed, you will have no problem. But I don't know. I haven't really tested it. OK. So now we are moved to a real case for RNA-seq. The brief background for this case is that in a very special ant species, we call the harpen netters, a worker, usually a worker, can take over the role of the queen under certain circumstances, while queen is missing. Okay. When this be triggered, there will be a set of process for a worker to be a queen, or we call it pseudo queen or Gamergate. There are terms for this. So this pseudo queen will have a different from original queen. We call it virgin queen. It's different. Have morphology changes and also function changes, such as laying eggs, producing the baby ants. This is a nature existing process, doesn't require any type of genetic code alteration. So we thought this is a very nice model for studying epigenetics in system level. The purpose of this study is to find out what genes are changed during this transition process. Okay. So first, we need to dissect this process. This process starts from the moment of sensing the world needs a new leader. And the ants became more active and crazy. Looks like this. And then change morphology and physiology fundamentals to adapt fat bodies and ovary. To lay eggs, need an ovary. And begin the process of dueling. Everybody has equal chance. Why you can be a virgin queen, why you can be a Gamergate, I can't. So they will fight each other. And finally, a winner will get a crown. This is a timeline for this process. So to understand this process solely, then you can do this experimental design and create analysis model. Because this process usually take a little bit of time to finish. And the lifespan of ant is not as mice or human. So along this transition process, there is a hidden factor that's aging. And this aging factor always comes with this transition process. So when you do this study, the genes you would like to know is along with the transition for a worker to be a queen. You absolutely need to exclude those aging affected genes. So analysis model here got to be much more complicated than a single factor experiment. Okay? So similar procedure we have FastQ file. We start from there. we do sequencing level control, the quality control. You do the trimming if necessary, and mapping to the proper genome. That's the first step you want to do. Then you do the data pre-processing. You get the BOM file conversion. You get BOM file sorted, and then you do the duplicate reads removing. As I said, to deal with the first issue. Then you do the reads assignment. Then you do data normalization and the sample level quality control. Okay? Then you do the gene selection. So after reads assignment and the normalization, you will end up with this data matrix. This is also a slide. I will tell you why gene length adjustment is unnecessary. Here in this table, you see all samples are column, all rows are genes. If you know this trick, to see if a gene differentially expressed between different samples, you know the calculation and the formula is actually compare the samples to samples. The formula doesn't compare genes to genes. Okay? So, which means same gene cross different samples will always have same exact length. In your formula, you do this calculation. If you put the gene length in, gene length adjustment in, actually length of that gene, that will be a denominator of the two sides of that equation. Later or sooner, that denominator will be removed. Why you do that at the beginning? So gene length adjustment for differential gene expression is totally unnecessary. Okay? However, after gene selection, if you want to do the good term or some functionality analysis, if those analyses will have multiple hypothesis testing involved in, then you need to do gene length adjustment. Okay, clear? So then you say, why I can't just do that at the beginning? I say, if you do that beginning, because gene length adjustment can bias to the short length of genes. Those short lengths of genes will be lifted during your multiple hypothesis testing. Those guys will get advantage. So your true genes, differentially expressed, will more or less be excluded from your list. Okay, with this table, normalized data, now we are going to do sample level control, sample level quality control. Nothing very special. You just do the principal component analysis to analyze variations within the groups and the variations between the samples. Here we see that these 3 months ant sort of separated from their group. I don't know what does this happen in the beginning, but this is clearly not a good sign. So I usually exclude this type of sample from analysis. Okay? Then you build your analysis model. In this case, I used a simplified two-factor mixed model to generate 4 formulas. For aging affected genes, I use model B, subtract model D. to get the transition-related genes, you use model B to subtract model C. For those interaction genes, which means genes perform differently in different age group or in different transition group, you use model A to subtract model B. That's basically calculation you need to do. Then you end up with this type of gene list. This gene list, I can tell the first gene list is aging-related genes, pure aging genes. These are basically transition-related genes. These genes really have something to do with aging and also with the transition. We call the interaction term. Okay. Here are the deliverable results from RNA-seq data analysis. You will get the FASTQ file and the quality control output, and also those processed data files. And also you will get sample level quality controls, and also those gene lists and the heat map of that select genes. And go terms if you want. Lots of time people just take those lists with them and do go term by themselves. Here is a summary of today's talk. So we have covered some general procedures for next generation data analysis. What can be delivered to biologists, and if there is advanced things you want to know, for example, more complicated data modeling and sophisticated analysis. Here, I would like to, I will end here by thank these guys, people from Danny Lab, Roberto, Juan, Wei, Philip. They provide lots of data. And from Yannis Lab, Thomas, Luisa, and Petri, and all my CHIBI colleagues. Thank you so much. Any questions? Yeah, go ahead.
So for barcode problem that you mentioned, so is it the Illumina protocol or your internal customers barcode, the data you use? You mentioned your barcode bias.
Yes, yeah.
So the data set you used, it's Illumina's protocol's barcode or your customized barcode?
It's Illumina barcode.
So
This is Illumina barcode, not Danny Lab developed barcode.
So is that a good barcode quality? I mean, do you see how many sequences has been identified?
How many sequences?
Yeah, I mean, has been demultiplexed. Is that good, or I mean, how good the data set you use? I just wonder why the barcode cannot work very well. I think that's very cool.
I don't know either. I want to know.
So I want to know if the barcode were sequenced very well. I mean, the quality of the barcode, how good they are.
I would say fairly good. It's pretty much as a non-barcoded sample. Nothing. You can see a very obvious difference between barcoded sample and a non-barcoded sample. This is not a unique case. Before last week, we have another set of samples from Yannis Lab, which also be barcoded. And Luisa, she has 2 previous batches which didn't have barcode. So when I plot those barcoded samples with non-barcoded samples, because all those from semi-exactly experiment are biological replicates, I do see a problem again. So I believe there's something going on with Illumina barcode. As I discussed with Danny and some other guys, just try to be safe, we need to do more tests. Okay. Illumina I called didn't really give me very useful information, and they told me that might be a lab preparation, sample preparation problem. You know, I can't really rule those variation out.
Is that the same number of total rates, or you have saturation problem? I mean, not thickness very well.
The barcode sample is just similar to the non-barcoded sample. It's nothing really different. So you don't see from file, at least you don't see this type of problem. Okay. Thank you all.